Welcome back, class. So today, we're going to start getting into nucleic acids and how nucleic acids are involved with DNA and RNA. So far we've really covered lipids and carbohydrates. Well, nucleic acids are going to be one of our other macromolecules. So when we look at nucleic acids, one of the big things that nucleic acids do, or their function, is really to store and transfer genetic information. And you'll see that we have these two classes of nucleic acids here. We have deoxyribonucleic acid and ribonucleic acid, also known as DNA and RNA. We'll talk more about DNA here in just a little bit, but when we look at RNA, and we look at both of these really put together, both of these are really polymers that are composed of a lot of nucleotide monomers. So again, both DNA and RNA. Now they do have some slight variations in their monomers, and we'll talk about that when we talk about the differences between the two. And then when we look at how they're put together, the monomers between the individual nucleic acids are covalently linked, okay? And they're done by utilizing phosphodiester bonds. And over here, I kind of stuck in a little bit of an image that you don't have in your notes, but you'll kind of see what the appearance of a phosphodiester bond actually is. You can see kind of this PO4 to the minus third. So again, this is kind of the idea of what a phosphodiester bond is, okay? You'll have the O here, bond with one pair of shared electrons to a phosphate, along with this O here also. So these are the ester bonds here and diester because there are two of them in each of these. So now when we look at a nucleotide and we look at the basis of an actual nucleotide monomer, we're always going to see three things with it. We're going to see a sugar, a phosphate group, and a nitrogenous base, okay? So when we look at the sugar is going to be typically a 5-carbon pentose sugar, okay? When we look at the phosphate group, you're going to see that it's kind of this PO4, and it's going to be attached at carbon 5 of the sugar, okay? So if we kind of go around, here's carbon 1, 2, 3, 4. Carbon 5 is kind of off of this additional methyl group, and so it's going to be connected to that number 5 carbon. And then we have the nitrogenous base, and it's going to be attached to the same sugar, but it's going to be attached there at carbon 1. So we have the sugar down here, the phosphate group, and then the nitrogenous base here at carbon 1. Now, these nitrogenous bases can either be single ring or double ring structures. We'll talk about each of those nitrogenous bases here in just a second. So when we look at the nitrogenous bases, there are 3 pyrimidines, which pyrimidines indicate a single ring base. So those are going to include cytosine, uracil, and thymine, okay? Now, there are also 2 purines, and purines are double ring bases. So basically they have 2 ring structures bound together, and that's going to include adenine and guanine. And then you'll notice that the nitrogenous bases within either group differ in the functional groups attached to the ring. So if we take a look at those nitrogenous bases, just to kind of get an idea. Again, here are the pyrimidines, here are the purines. When we look at the pyrimidines, again, these are single ring structures. You can see one individual ring, and the same thing on thymine. Here we have the purines. The purines have 2 individual rings, okay? So these are double ring structures, adenine and guanine. The pyrimidines are single ring structures. Now, when we look at the locations of both of the, or all of these, with the purines, those are going to be found in both DNA and RNA, okay? When we look at the pyrimidines, cytosine is found in both DNA and RNA. But now when we look at uracil and thymine, there are some differences. Uracil is going to be found in RNA only. Thymine is going to be found in DNA only. So to kind of simplify this a little bit, we usually indicate uh, these bases based on their first letters. We usually call them A, C, T, G, and U. So when we do that, if we look at uh, DNA, DNA is going to include A, T, C, and G. When we look at RNA, however, it's slightly different. It involves a U, C, and G. So there are a couple differences in that, and those are the bases that you're going to want to make sure that you know and understand as far as where they're located, in which macromolecule, whether it's RNA or DNA. So if we start to look specifically then at DNA, that's going to be kind of the first of our nucleic acid macromolecules here. So when we look at this, we look at DNA, it is a double-stranded nucleic acid. So it basically has two strands of nucleic acids on either side. And so what it looks like is this kind of spiraling staircase. When we look at DNA, it's going to be located in the chromosomes in the nucleus, but also in the mitochondria, okay? Now, when we look at the DNA molecule, again, we're looking at a sugar, a phosphate, and a base. Well, our sugar is going to be deoxyribose. Our phosphate group obviously is the same. And then we utilize one of the four nitrogenous bases that are involved with DNA. And that's going to be adenine, guanine, cytosine, or thymine. Again, there is no uracil found in DNA. Now, the double strands, because again, we have a strand on either side, those are actually going to be held together by hydrogen bonds. Now, remember going back to earlier, hydrogen bonds by themselves are very weak. But collectively, when you put them all together and start giving a structure, in particular, in this case, a three-dimensional structure, those hydrogen bonds can actually become very strong. And those hydrogen bonds then are formed between the complementary bases. And so you'll want to, this is going to be one of those things that you do kind of want to make sure that you understand fairly well. Is that when you look at the pairing of them, thymine is always going to be paired with adenine. Guanine is always going to be paired with cytosine. So A to T, G to C. Now, with RNA, RNA's got some slightly different characteristics. It's not double-stranded, it's only single-stranded. It's going to be located within the nucleus of the cell, but then we're also going to find sections of it in the cytoplasm of the cell, especially when we're getting into protein synthesis. When we look at the composition of it, again, we have a sugar, a phosphate, and a base. Well, in this case, we have ribose is our sugar. We still have the phosphate group, and then we still also have one of our four nitrogenous bases. However, notice the difference. A, C, and G are still there. T is no longer there, and T is now replaced by uracil. And so there is no thymine here. So again, you want to make sure that we understand, again, what bases are involved with RNA and DNA. So when we look at RNA and DNA and we kind of look at them, in graphical representation, we are basically looking at a situation. Here is DNA. You'll notice these kind of two ribbons. These are the two strands of DNA. Here we indicate our nitrogenous bases. We have T and A together. We have C and G together. Here on the side, you can see how they're linked together by phosphodiester bonds. And then here you can see where they are bound together by the hydrogen bonding of hydrogen and oxygen. On this side, you can see where we have just RNA. This is a single-stranded structure. And you can also see, again, the same types of things. You can see the phosphodiester bonds here. You can see that in this case, we no longer have the thymine, we have uracil. So here are our nitrogenous bases. Here's your ribose sugar, which you'll notice is there. This, in this case, is deoxyribose sugar. and then there's our phosphate group. So that is the structure of RNA and DNA. Now, if we look at what ATP is and we look at adenosine, okay, remember adenosine, as far as the nitrogenous base, is adenine having the nitrogenous base group. Here's our ribose sugar, and then here's our phosphate group. So in this case, we're looking at adenosine triphosphate. So the nucleotide is composed of the nitrogenous base adenine, a ribose sugar, and 3 phosphate groups, okay? And at least in this case with ATP. The central molecule in transfer of chemical energy within the cell is this ATP molecule. And the reason being is that when we look at these squiggly lines here, these squiggly lines really are indicating very high energy bonds. And so you'll see that we have these 2 covalent bonds between these last 2 phosphate groups, and they are, again, very energy rich. And energy is released when these particular bonds are broken, okay? So again, very high energy bonds in this triphosphate group, which is really what gives us the triphosphate here of the adenosine structure right here. So now what are some of the functions of proteins? Well, proteins, one, are going to serve as catalysts. So we typically refer to them as enzymes, things that speed up reactions, in particular metabolic reactions that we find within our bodies. They can also act in defense. They can aid in transport by getting things into and out of cells. They can contribute to structural support, in which we'll find a lot of these proteins within our plasma membranes. We can use them for movement, in which we have a lot of different motor molecules that are involved with that. They can perform regulation as well as provide storage. So a lot of different functions of proteins. So if we look at the general protein structure then, proteins are really made of one or more strands of amino acid monomers. Now we'll talk about amino acids here in just a little bit, but amino acids are really a collection of 20 different structures that are found within living organisms, okay? So there are 20 different types of amino acids. Each amino acid, as you can pretty much tell by its name here, amino acid, has an amine group and a carboxyl or carboxylic acid functional group. And both of these are covalently linked to the same carbon atom. Now, we also will find that carbon is also covalently bonded to a hydrogen and a different side chain structures. We are going to reference these or call these R groups. Now, these R groups obviously will have names, but it's these R groups, these kind of structural groups that distinguish different amino acids from one another. When we look at the amino acids, they're all going to be covalently linked by peptide bonds. That's kind of a specific name that we call or that we reference to amino acids. And these peptide bonds are going to be formed during dehydration synthesis between the amine group of one amino acid and the carboxylic acid group of another. So what we see is that a hydrogen is going to be lost from the amine group, and an OH here is going to be lost from the carboxylic acid group of another amino acid. And by joining these two structures together, we are going through this dehydration synthesis reaction where, again, we're basically pulling water out of this particular reaction and out of this system. When we look at the amino acid structure, we have a couple different things. We have an N-terminal N, which has a free amine group, and a C-terminal N, which has a free carboxyl group. So if we kind of take a look at some graphical representations here, here in A, we have the amino acid structure. Again, here's our one carbon with a hydrogen covalent bond to it. Here's that amine group, okay? Here's the carboxylic acid group. Again, both of them covalently bonded to the same carbon atom. Then we have this last one down here. And this last structure is our R group. Now again, this R group can represent a lot of different things. It can represent something as simple as a hydrogen. It can represent something as simple as a methyl group, which is another CH3 group. So there's a lot of different structures that we can add down there. And remember, we have 20 different amino acids. When we look at the peptide bond and we actually form a bond between individual amino acids, again, here's the amine group of one. Here's the carboxylic acid of another. The amine group of one, carboxylic acid group of another. And when we start adding them together or getting them to bond with each other, again, you'll notice that we take the hydrogen of the amine and the carboxylic acid, or the OH, I should say, of the carboxylic acid group. Those bond together, we give off water, and then you'll see that we end up having a bond here between carbon and nitrogen. And so now you can see what a protein looks like as we take a series of amino acids and put them together. And obviously every amino acid has its R group. We start here with the N-terminal N. Here we have our C-terminal N. And each of these here is a peptide bond bound or formed between individual amino acids. So if we look at the general protein structure in certain strands of amino acids, we can look at an oligopeptide, which normally is going to be between 3 and 20 amino acids. We can have a polypeptide, which is between 21 and 199 amino acids. And then we have a protein, and proteins are these really long structures that are made up of more than 200 amino acids. And then we have structures called glycoproteins. So glycoproteins are proteins, so they have more than 200 amino acids, but they have some sort of carbohydrate, therefore kind of the glyco component of it. And so we'll see a lot of glycoproteins on erythrocytes, and this helps us in determining the different ABO blood types or blood groups, okay? So let's look at a few of our amino acids. Before we actually get into amino acids, let's use this in particular as part of our attendance for week 3. So again, you've heard me talk about our code words, just different words or things that either are material based or sometimes they're just random outside of class type of things. But this is going to be our code word. It's going to be amino acids. And so once I get our attendance sheet put, our attendance quiz put up, that's how you'll go in and take your attendance quiz. Again, it'll be for all of the lectures within week 3. So don't try to take it the first time and then wait. Try to get your code words down. Just remember them. Again, they're just a couple words or single words. But in this case, it's amino acid. Okay. So if we also then look at this, we start to organize them based on their R group. And so when we look at amino acids and we look at nonpolar amino acids, nonpolar R groups are going to be or contain hydrogen or hydrocarbons. They're not going to have a lot of other elements to them, okay? They're going to be grouped typically with other nonpolar amino acids by hydrophobic interactions. Now, we also then have polar amino acids, and their R group typically is going to contain other elements besides hydrogen or carbon. And because they're polarized, they are actually going to form interactions with other polar amino acids and with water. So again, I'm not sure necessarily how easy this is for you to see this chart. I'm going to try something here and I can't zoom in. So if we look at this, again, you should have this in your book, so it'll be a little bit easier for you to see. You'll see that here we have kind of a few non-polar molecules. Here we have some polar molecules. And so you can kind of go through and review some of those. We're not necessarily worried about making sure that we know every single amino acid that there is, so much as you just really understand the basic structure of what an amino acid is. So as we start looking then at how we utilize amino acids, and we get to their sequence in protein conformation in developing a protein and creating one, we have to look at the levels of structure in order to get to that point. And so there are 4 levels of structure in protein formation. The first one is primary structure. And primary structure is nothing more than the linear sequence of amino acids. And so each of these represents a different, each of these bubbles or balls here indicates or represents an individual amino acid. And so you can see as we blow it up then, again, here's an R group, indicates one amino acid. Here's an R group. In this case, we'll say it represents a different amino acid. And you'll see how the carbon of the carboxylic acid group binds to the amine group of the next amino acid. And here again, we form our peptide bond. And so we do this throughout the entire length of the chain. Remember, in proteins, they tend to be upwards of over 200 amino acids bound together. So we just have this complete series of amino acid sequence put together. That then leads us to secondary structure. So now with secondary structure, we're actually going to start to see a little bit more of a 3D conformation. We're going to start to see a little structure to it. And so when we look at secondary structure, there are really 2 types of characteristics that we see within secondary structure. The 2 types are going to be, one, an alpha helix. And so you'll see a helix here basically has this spiral shape. And we also have a beta pleated sheet. And in the beta sheet, these are a little bit flatter. They kind of go up and down a little bit, but they are flattened or relatively flattened structures. And so as we start to get into this conformation, you're going to see that these patterns are going to repeat themselves. We're going to have an alpha helices, and then we may have a beta pleated sheet, and we're going to have another alpha Cs and a beta pleated sheet, and so on and so forth, in order to get the structure that a particular protein needs. And so in here then, you at least can see where we have alpha helices, and then we have a couple beta pleated sheets, and then some more alpha helices and beta pleated sheets. So you can see that the structure from the secondary structure perspective is at least kind of laid out. So now we get to the tertiary structure. And with tertiary structure, we now start to form a true 3-dimensional shape of an individual polypeptide chain. Within tertiary structure, we have 2 different types or 2 different categories. We can have a globular protein, and that's actually going to fold itself into a compact shape. And you can see we have multiple secondary structure levels here kind of strewn throughout. And so they basically start to compact themselves onto one another to develop some form of a 3-dimensional protein. We also can look at the fibrous protein, and these tend to be a little bit more extended linear type molecules. Okay, they're a little bit more fibrous or strand-like. And then lastly, we get to quaternary structure. Quaternary structure is going to be present in proteins with 2 or more polypeptide chains. Okay, and so what we're going to see, I'm not sure why I still have a quaternary structure graphic there, but what we're going to see is we're actually going to take a globular protein here. We'll take this structure, combine it with another structure, and so we actually will then have 2 globular proteins put together, or several in some cases. And by doing that, that actually forms us a functional protein. So for example, hemoglobin is actually 4 individual polypeptide chains. So take this globular protein, take another globular protein, and basically take 4 of them and put them together. And that gives us hemoglobin. Okay? So that gives us a functional protein in which we have multiple polypeptide chains put together. So again, I apologize if this still says tertiary structure. Um, I'm not sure I actually have the quaternary structure. So make sure that you check your book for the quaternary structure graphic up there. Now, as we look at amino acids then, uh, amino acids obviously can be added together to form a protein. Well, when we look at proteins, they can also be broken down. Oftentimes, these are due to extreme uh, environmental conditions, but we do see different change or different changes to proteins. And this can occur from a conformational perspective and how they are laid out and how they're developed. And what happens is when we denature or break down a protein, it actually disturbs the activity of a protein. Now, normally when we do this, it's usually irreversible. And so what we typically see is we see, for example, an increase in temperature or an increase in acidity. Both of those have the potential to damage uh, proteins within the body. So if we think about this, um, obviously I'm assuming everybody understands that an egg, the egg white is made up of a lot of protein. Well, when you throw it in a skillet, you're obviously increasing temperature quite significantly. And so when you do that, you'll notice that the egg white, which is normally clear when it's raw and the proteins are good, actually becomes white when some of those proteins have basically been broken down. And obviously we can say, hey, once we grill the egg or throw it in the skillet, once it's cooked, it's not like we can go back and change it back. And so you see how essentially uh, denaturing some of the proteins is usually irreversible there. And again, that's just one example of increasing the temperature, okay? So what are some other causes of denaturation? Obviously, temperature was one of them. pH is another one. And pH changes can interfere with the electrostatic interactions, as well as some other intramolecular bonds. So we talked about what the pH of blood was earlier when we actually discussed pH. And so the body really has a very small range that the pH needs to be in order to function correctly. Anything outside that range is where we really start to see some damage to other tissues. And this is just kind of one of them. Increase hydronium or decreased pH actually causes some of these electrostatic interactions really to start to break apart. And so we add hydronium ions. So now instead of it bonding and forming a bond with, in this case, the opposite amine side, now we actually can break that bond, and now the hydronium ion comes in and actually forms right to the oxygen, which is what the oxygen wanted to begin with. So again, that's what we start to see then with too much hydronium ions in solution, okay? And so that'll be it for this part of chapter 2. This is not necessarily a full lecture, uh, but I will start to get into chapter 3 for you to start watching. And again, we may end up having 3 lectures that actually show up um, within week 3. But again, it's all just the stuff that you really need to get accomplished within that particular week. So otherwise, we will see you into chapter 3.